Hi, thanks very much for the invitation to speak today. The Deep Mutational Scanning Symposium has become one of my favorite yearly activities here, and so I'm really glad to be able to participate. Today, I want to tell you about a project that's been an ongoing collaboration with my lab, Doug's lab, Alan Reddy's lab, and Debbie Nickerson's lab, and that's to use deep mutational scanning to try to functionalize variants in pharmacogenes. And my particular emphasis in this project has been using yeast activity assays as part of this effort. So of course, we all know that the problem in modern genetics is that we've sequenced lots of things, but the amount of things that we've annotated is much, much smaller. And so how are we going to meet the challenge of that? And of course, to this audience, the options are obvious. We need to come up with high throughput functional assays that will let us turn these variants of uncertain significance into variants that we can give some sort of clinical recommendation on the basis of. And there are a few different approaches to this, many of which you'll hear about at this symposium, which is really exciting, all the variety. The approach that my lab has taken is to take our expertise in using yeast as a system for high throughput genetic screens and to apply that to human genetics, which of course has been something that yeast geneticists have been doing for decades, but not at this scale. So there are a few different ways you can do this. You can take advantage of evolution and homology, and you can make mutations in the yeast gene that are homologous to the sites in the human patients. And if you're interested in this as an approach, we have a kind of a shallow mutational scan that we did for mismatch repair gene, MSH2. And I have to say, the part of the collaboration of this actually grew out of one of the deep mutational scanning symposia, where Anya gave a talk about this project and Brian Schertz was in the same section. And we ended up having a collaboration that you can read about in this preprint. You can also use cross-species complementation, where homology goes even one step further to functional homology, where you can take the human gene, replace the knocked out yeast gene, and maintain its function in the yeast cell. And then you can make your mutations right in the human gene. If you're interested in learning more about this approach, Renee Geck in my lab has a poster about using this to assay G6PD mutations, glucose 6-phosphate dehydrogenase, which is another important pharmacogene. And finally, you can use just heterologous expression, where any protein that you can express in yeast that works, you can essentially use yeast as a little biochemical test tube in order to study its function. And that's the emphasis of today's talk, where we're developing assays to use yeast to measure cytochrome P450 activities in mass. Okay, so the particular application that I'm going to talk about is pharmacogenomics. And this is a really fascinating area because the concept here is that using your genotype, we should be able to figure out what drug dosage and what drug type should be the best for you and let you avoid abnormal drug reactions, which cause all sorts of bad outcomes for people, both in terms of drugs that don't work as appreciated, but also in terms of adverse reactions. And these genes, since they're involved in processing chemicals, are extremely variable, which is really fun for a geneticist who works on evolution like me. There's a huge amount of variation in these genes, and so nearly everybody has some sort of a variant somewhere that is clinically actionable. And these are actionable, right? The nice thing about these genes is that you can use the results right away to try to make better guesses about a starting place. Obviously, you still need medical monitoring, but if we can avoid some adverse drug reactions by starting out with a higher dose, for example, then you don't have to do the experiment on the person in as many of the cases. Okay, so here's just one example. It's a classic, right? Warfarin is one of these drugs where the clinical amount of drug that you deliver is actually pretty narrow, and it depends quite a bit on genotype. And there are some variants in a gene called VCOR that cause resistance, and Doug's lab actually has a really beautiful deep mutational scan of VCOR that they recently published. And there are mutations in the CYP genes that cause other phenotypes as well. And so this is something that these genes is just one example of a drug that is metabolized by these genes. Cytochrome P450s are this huge family of enzymes that do all sorts of amazing things. And they're also involved in metabolism of majority of drugs. So they make a really compelling target for some of these strategies. Because they're such a big family, this is one argument for using yeast as a system for them, because you can get some background thanks to the overlapping specificity of these enzymes in, say, liver cells. So by transplanting them one at a time into yeast cells, which have cytochrome P450s, but not ones that overlap in specificity, we can hopefully do this kind of high throughput screening that is demanded by the large number of variants in these genes. Okay, so the gene that I'm going to talk about today is CYP2C9. We also have a few other CYPs in the pipeline, but this is the one that's made it the furthest. And this is an appealing target because it's an important clinical gene, obviously. That's one of the ones involved in warfarin resistance and sensitivity. But there's a ton of mutations, hundreds of mutations that are have been discovered, but have very little annotation information. And frequently that's because they're rare. And so it's hard to do any kind of association tests and things like that. So we have a few options for annotating these, right? You can test them. That's historically what's been done. And this can be very accurate, but of course it requires finding a variant, constructing it, and then testing it. Computational predictors are getting better and better, and hopefully, in fact, being trained on deep mutational scanning data will get them to the point where they need to be in order to be accurate enough to be able to be used for this purpose, but they're not quite there yet. So deep mutational scanning is, of course, the solution that we turn to. Okay, so all the steps involved in this are pretty much the same as what you'll see all day today, right? So we're making a library. In this case, the gene is 490 codons, which is pretty big. And so we tried to do the entire thing and got pretty close. We constructed a yeast strain that was really good at expressing active cytochrome P450s. We came up with an activity-dependent labeling scheme that I'll tell you more about, but it essentially turns cytochrome P450 activity into a fluorescent signal that we can then sort on. And then we can take the sequences of the particular variants in these different bins and use that to convert into an activity score. Okay. So I'm going to skip over the details about all of this part. In fact, we'd probably just purchase this library today, but it took a tremendous amount of effort on the part of Elisa Wong in my lab and Clara Amorosi, and also with help from Kate Sitko and Doug's lab. But we got there. And the other part of this that I will talk a little bit about was building a super strain of yeast that is really good at expressing these. And the modifications that we ended up with were expressing 2 cofactors for the CYP, and also knocking out the proteases, which is an old protein expression trick, and fixing some of the lab strain deficiencies that the strain has accumulated. So at the end of the day, we were able to get high expression, and we're able to get good activity out of these cells. Okay, so how are we going to do this activity assay then? So this was an idea that Doug brought to the team, and that has worked really well, and that I think actually has a ton of promise for other types of enzymatic assays, and not just yeast, but in other cells too. And that's activity-based protein profiling. So the idea here is that you have some sort of a probe that can interact with the enzyme in an activity-based manner. And so in this case, we have a probe that when it reacts with an active CYP will covalently attach to it. And at the end of that probe, there's a click handle so that we can click on a fluor. And that fluor is what we eventually detect. And so this had a few different challenges. First of all, it had some nice things to it, which was that it's activity dependent and it's covalent, so it doesn't leak back out of the cell. And there was some proof of concept already from Aaron Wright's lab, which was great. And they were actually able to share with us some of their universal probes that they'd developed as a starting place. However, this hadn't been shown to work with yeast yet. We had to see, can we get all this stuff in there? Can we get the click chemistry to work in yeast? And do we get enough labeling? Because the stoichiometry of this did not seem to be totally working in our favor. The cat has decided to come visit. I'm sorry for the interruption. Probably there's a bingo card out there with that on it. So congratulations to you. Okay, so we ended up doing this using not one of the universal probes, but using a probe that was a little bit closer to one of the real substrates of this enzyme. And so that's tenelic acid. And this is where working with Alan Reddy's lab was really great, because Matt McDonald was able to synthesize these for us. So here's what the flow data look like. And I want to point out just real quick that these are plasmid-borne variants. And so some portion of the culture actually loses the plasmid during the experiment. And so some of these low values are false in the sense that they represent cells without a plasmid. Since they don't contribute to the sequencing, it doesn't matter, but it does make the FACS data look a little messier. Okay, but here's what wild type looks like. We get this nice distribution here. we tested a few of these highly studied alleles that have really known consequences. And so this is one called the star2 allele, which has a reduction in activity. And sure enough, we see a shift to lower fluorescence there. Here is an even lower still star allele, star3. And then we have this catalytic dead mutant, which has essentially no activity. So this really was promising to us that we were able to measure CYP activity using this click assay. Okay, so that emboldened us to go for the full library. So here's the distribution of the library, and you can see it ranges from wild type like to null like. So what we did was we binned it according to 2 bins representing the highest 10% and the next 10%. Another medium bin, and then all of these lower ones, which represent both loss of function mutations, but also those that have lost their plasmid. We sequenced them and used the scores of the synonymous mutations and the nonsense mutations to kind of bracket the score range, as is typical for these experiments. Okay, so what did we find? Well, if we look at the entire distribution here, here are the synonymous alleles, and they have, since we set their scores at 1, that's what we ended up getting. And so, of course, the nonsense alleles, we set their score at 0, so that's also what we got. But what was really great to see was this really tight distribution in both cases. And now here are all the missense alleles that were in our library. And so they really range. And if we go and ask, okay, now where are those control alleles that we already knew about? Well, they end up in exactly the right order, which is great. So here's star2, here's star3, and here's the catalytic dead. And that represents really pretty closely what we got on the single strain one at a time flow cytometry data. So we also wanted to compare this against one of the gold standards in the field. And so Matt in Alan's lab was able to do a mass spec assay that asks about turnover from a warfarin into one of its catalytic, if CYP acts upon it, here's what the outcome is. And so we got pretty good correlation between the warfarin activity and our activity score in the click-seq assay, which is what we're calling it, click-seq. You can see that there are a few things that are kind of not one-to-one here. And so we are investigating whether some of those are somewhat substrate dependent, for example, or might represent other kinds of artifacts that we possibly could see in our assay. But in general, we were pretty pleased with this result. Okay, so one big question though, this tells you that things have low activity, but it doesn't tell you why. It doesn't tell you kind of a molecular mechanism for what that loss of activity is all about. And in Doug's lab, Melissa performed a parallel assay, which is a VAMP-seq assay to look at abundance of these protein variants. And this allowed us to do 2 things. One, it allowed us to check things out in a human cell culture, which really made us more comfortable about whether the yeast were actually giving the right result or not. And then it also allowed us to kind of put a mechanism on some of these. So VAMP-seq, just in case I'm the first one to talk about this, is essentially a method where you fuse your protein of interest to GFP, and then you ask how much fluorescence, again, the same kind of outcome as with the click-seq assay, but now assaying a different feature of the protein. So while our yeast assay measures both activity and abundance, a combination of those 2, this really is more focused on trying to study abundance. So what did we get? Well, interestingly, if we correlate the abundance score with the activity score, what we get is a range down here where essentially if you're below a certain threshold of abundance, you have very, very low activity. And if you're above that threshold, then there's a correlation between abundance and activity. But of course, that correlation is not perfect. And so that's one interesting reason, I think, to do both, is that there are certainly variants here which have high abundance scores, but have low activity scores. And so if you had only done VAMP-seq on these, and in fact, there's another study that did VAMP-seq on a small number of previously observed CYP2C9 alleles, then you would miss out on these alleles that are potentially important, but are not assayed by just abundance alone. So we think the combination of these 2 things is pretty nice and pretty powerful. If you turn them into a heat map, so you can now see the effects spread out over the length of the protein here, where dark blue is a low activity. We didn't see actually any very high activity alleles here, so which is not a surprise from the CYP literature. They might be out there. We're still looking for them, but not so relevant for this particular study. But you can see that in general, regions that are in these important helices up here often have decreases in both activity and abundance. But there are some regions that are only different in one or the other assay, which is kind of interesting that certain regions of the protein have these different features. Overall, we were able to get at least one score for over 8,000 variants, and we have both scores for over 4,000. So that gives us a really great data set. The CYP enzymology structure function people, I'm hoping will really have a great time looking more in detail at our data set to find out features of CYP biology that maybe have been underappreciated. And I should say that there's more on that in the preprint, including a lot of nice structures that have things mapped onto them if you're interested in that. But with my remaining time, I want to get back to where I started, which was, can we turn these scores into something that's clinically relevant and that could be used to help patients? And so what we did was we turned these scores that we got from our activity assay into categories that kind of match what the databases use for deciding if there's activity or not. And these activity assays, when we bin them and look at where their status corresponds with what's in the databases, and in particular in the CPIC database, that this consortium is essentially responsible for curating all of the data on variant information and deciding what reaches a evidence threshold that's worthy of being able to give a recommendation to clinicians. Here's what it looks like when you use activity classes, and here's what it looks like when you use abundance classes. And what we see is that in general, the alleles with no function we call as no function. And the alleles that have decreased function, we generally call as decreased function, although there are a few that are edge cases here. You can see that having both assays together is helpful for things which are decreased function, but not necessarily because of their abundance. So when we map this now onto all the known variants in Nomad, so this is hundreds of variants, and they're mapped here according to their allele frequency, just to emphasize that not all of these are rare. The ones that already have a star allele named by them, those are the ones that have been assayed to some degree, but a lot of these have not. And so here, for example, is one that we call as a decreased function that would probably be worthwhile to tell a patient about. And yet, and a lot of people carry it, and yet there was no previous information about it. So there's this one and 340 more like it. And so a lot of these had absolutely zero functional guidance, and a whole lot of them have decreased activity, including about a 1/10 of them that have total loss of function. So we think that this is an important contribution, and we're now in the process of talking to the databases about how best to convey it to their audience of both researchers and clinicians. Okay, so this is a project that we're still working on and are now pushing forward in different directions. Like I mentioned at the beginning, Clara's preprint is up, and we'd love to hear feedback and comments and if anybody reads it and has thoughts about it. We are in the process of doing deep mutational scans of other pharmacogenes. Soyeon, a new graduate student in my lab, is doing 2D6. And Renee, postdoc, is doing G6PD. And like I said, she has a poster if you'd like to learn more about that. We're interested in this concept of whether some of these variants might have substrate-specific effects. This is a shaggy dog story that I didn't have time to go into along the course of this, but we actually discovered this sake strain that turns out to have exceptional CYP expression activity. And so we'd like to explore that a little more to see if there are even more improvements we could make to the yeast engineering side of things. And of course, finally, figuring out the database side of this and how to interface with the human geneticist and the clinicians is something that's been a huge learning curve for me as a yeast geneticist for sure, but something that is important to do and that we are really on the way to making some progress for, hopefully. Okay, and so with that, I'd like to thank my lab in the, the now obligatory Zoom lab photo. In particular, most of this work was Clara's. Clara's really led this project from the beginning, and she worked very closely with Elisa, who is not in this photo because she's already moved on to a great biotech company. But between the two of them, they really did most of this work that I showed, and besides the VAMP-seq work, which was the work of Melissa in Doug's lab. And it would not have been possible without Alan, who's the card-carrying pharmacogenetics person who really has has taught me everything I know pretty much about cytochrome P450s, and Debbie, of course, the matchmaker of the whole project, who put the dream team together. So I hope that leaves some time for questions, and thanks again for the invitation.